Question number 116. Out of the 7 characters of P studied by Mendel, genes for 3 were situated on chromosome number 4. That for flower position, pod shape and plant height. Suppose if they were closely situated and were linked, then which of the following rule would not have been discovered or explained by Mendel? First option, law of dominance. Second, law of segregation. Third, law of independent assortment. Fourth, law of purity of gametes. Mendel was very lucky that he did not find linkage. Otherwise, he would not have been discovered a rule called law of independent assortment. How? Let's take all the options one by one. What is law of dominance? Law of dominance says that in a dissimilar pair of factors, you must be familiar with this, capital T and small t. When this kind of condition is present in a plant, when there is a dissimilar pair of factors, only one of the factors is expressed to itself, and this is called dominant factor, and the other one which cannot express is called recessive one. So this is called law of dominance. This recessive factors can express itself only in the homozygous condition, that is small t, small t. So this option is ruled out. Second, law of segregation. What is law of segregation? Law of segregation says that suppose if a diploid plant has 2 alleles for a character, let's say capital A and small a, then these 2 alleles, capital A and small a, can segregate independent of each other. Capital A can go in one gamete and small a can go in the another one. So these alleles can separate or segregate independently of each other. So this ensures the purity of gamete, but it can be possible in linkage too. So this option is also ruled out, and the fourth one, which is called law of purity of gametes, it is related to law of segregation only. So this is also ruled out. Now the remaining option is law of independent assortment. What does this law says? Law of independent assortment says that when there are 2 genes, suppose capital A and small a, these are the 2 alleles of a particular gene, and the capital B and the small b, it is the different gene. It has 2 alleles, capital B and the small b. These 2 alleles can independently segregate from each other. This was the law of independent assortment. For example, if A controls the seed color and B seed shape, then these 2 genes can independently separate from each other. A gene can separate independently from B, and this is called law of independent assortment. But if Mendel would have been discovered the linkage, then A gene cannot move independent to B. They will move together, and hence he could not find the law of independent assortment. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 117. Which of the given statements is true regarding Mendel's genetics experiments? First option, Mendel conducted his experiments from 1856 to 1876. Second, he performed his experiments on hawkweed, lublub and garden pea, but succeeded only with garden pea. Third, Mendel selected 7 true breeding pea plant varieties. Fourth, for the first time, Mendel used the term pure line. So select the true statement about the Mendel's genetic experiment. Mendel, he is the father of genetics. He conducted his experiments for 7 years, that is from 1856 to 1863. Hence this statement is wrong. Mendel selected 7 true breeding pea plant varieties. Mendel selected 7 characters. It means he took 14 true breeding pea plant varieties. So it should be 14. If for a character he took two varieties. How? One for tall and the next plant for dwarf. So as he took 7 characters, then he took 14 pea plant varieties. So this statement is also wrong. Fourth, for the first time, Mendel used the term pure line. No. For the first time, Johansen used the term pure line. So this statement is also wrong. Now see the second one. Mendel performed his experiments on hawkweed, lublub and garden pea, but succeeded only with the garden pea. Yes, it is true. Many of you may be knowing about it that he performed many experiments on many different plants but succeeded only in the garden pea, that is Pisum sativum, which you eat daily in your vegetables. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 118. In how many of the following organisms sperm will decide the sex of the progenies? A. Grasshopper, B. Drosophila, C. Birds, D. Butterflies, E. Moths, F. Humans. First option 4, second 3, third 2, fourth 1. Who decides the sex of the child in humans? You must say it is the male decides sex of the progeny or the child in the humans. Why? Because your answer is based on the male heterogamety. If a male is heterogametic, then male will decide the sex of the progeny because it can form 2 types of sperms. Whereas if a female is heterogametic, then a female can decide the sex of the child because it can have 2 types of eggs. So you have to choose the organisms in which males are heterogametic because then they can produce 2 types of sperms and they can decide the sex of the progenies. Drosophila, yes, males are heterogametic. Humans, yes, males are heterogametic. Grasshoppers, yes, males are heterogametic. In all these organisms, males are heterogametic and then they decide the sex of the progenies. In butterflies, birds and moths, females are heterogametic, hence eggs decide the sex of the progenies. So the correct option for the given question is option number 2. Question number 119. Select the option with correct set of statements for Punnett square. A. Developed by a British geneticist. B. Used to calculate the probability of genotypes of offsprings. C. Useful for monohybrid crosses only. D. It is a mathematical formula which helps to calculate the types of gametes. First option A, B and C only. Second A and D only. Third B and D only. Fourth A and B only. What is Punnett square? Punnett square is a graphical representation which is used to calculate the probability of all possible genotypes of offsprings. So this is true and it was developed by a British geneticist. So it is also true. C. Useful for monohybrid crosses only. No, it can be used for dihybrid, trihybrid crosses too. So it is not useful for monohybrid crosses only. So this is wrong. D. It is a mathematical formula which helps to calculate the types of gametes. No, it is not a formula. It is a diagrammatic representation which is used to calculate the probability of all possible types of genotypes of offsprings. So the mathematical formula which helps you to calculate the types of gametes is 2 raised to the power n, where n is a number of heterozygotes. For example, if this is the genotype of an individual, then you can calculate the types of gametes by applying the formula 2 raised to the power n. How many heterozygous pair are present? 1, 2, and both are homozygous here, so it will be not considered. So 2 raised to the power 2, that is 4. So the correct option for the given question is option number 4. Let's move to the next question. Question number 120. Calculate the probability of obtaining a zygote with the genotype capital A, small a, capital B, small c, capital D, capital D. if a cross is made between individuals whose genotypes are small a, capital B, small c, capital D, and capital A, small b, capital C, capital D, respectively. First option 1 by 2, second 1 by 4, third is 0, fourth 1 by 8. It is very interesting question. I really liked it. See the genotype of the zygote. It is capital A, small a, capital B, small c, capital D. And the genotype of the parents is small a, capital B, small c, capital D. This is the genotype of a first parent, and then the genotype of the second parent is capital A, small b, capital C, capital D. So if a cross is made between two parents, then what is the probability of getting this type of zygote? It is very easy question. You don't have to calculate anything. Just put your brain over here. If zygote contains capital B, one capital B should come from the parent 1 and one capital B should come from the parent 2. Parent 1 can contribute this capital B, but parent 2 does not have capital B, hence it cannot contribute this capital B to the zygote. Hence there will be no possibility, no probability of getting this type of zygote from these parents. So the probability is absolutely 0. So the correct option for the given question is option number 3. Let's move on to the next question.